My name is Bob Harris, and I am a professor of immunotherapy and neurological diseases at the Department of Clinical Neuroscience, Karolinska Institutet. My research group, the Applied Immunology and Immunotherapy Group, is based at the Center for Molecular Medicine at Karolinska Hospital in Solna. Our research activities center on understanding the immunological processes that characterize neurodegenerative diseases such as multiple sclerosis, Alzheimer's disease, and ALS. Having this knowledge is vital for the more important activities of developing novel immunotherapies, which we test in experimental models of these diseases. We have a long-term goal of introducing these into the clinical treatment of patients. Here follows a short summary of our most recent study that was published in Nature Immunology. It is the brainchild and primarily hard work of lead author and my PhD student Harold Lund, together with colleagues in a collaborative group effort. In the homeostatic state, monocytes that circulate in the blood are separated from the central nervous system, the CNS, by the blood-brain barrier. The macrophage cells resident inside the CNS are called microglia. The origin of these two populations of myeloid cells is different, monocytes being derived from progenitors in the bone marrow, while microglia arise from yolk sac progenitors and migrate into the CNS early during development. This means that they are not the same cells, even though they look quite similar in morphology. We have been interested in studying what happens when microglia are genetically deleted. In the most commonly used systems, this has been studied in a steady state, that means in a non-disease situation, and the loss of microglia is usually followed by a quick proliferation of the remaining cells or progenitors, leading to a repopulation of the CNS niche with bona fide microglia. We were interested in more long-term depletion of microglia, and so have developed a system to achieve this, with diphtheria toxin being expressed inside microglia under control of the CX3CR1 gene. When tamoxifen is given to such mice, then the diphtheria toxin is released within the cell and it dies. In this situation, some microglia survive, but they cannot repopulate the CNS niche efficiently. The CNS does not like to be without myeloid cells, so it permits the infiltration of monocytes from the blood. Once inside the CNS, these adapt to the CNS environment and become microglia-like with respect to the proteins they express and their functionality, here shown as a macrophage. The research hypothesis we tested was that the ability to respond to the cytokine TGF-beta was crucial for this enforced repopulation. We thus genetically engineered mice in which the infiltrating monocytes would not be able to respond to TGF-beta as they lacked the receptor. Application of tamoxifen to these chimeric mice resulted in both microglia depletion, which initiated monocyte infiltration into the CNS, as well as inhibition of the TGF-beta expression on these monocytes. While control chimeric mice remained healthy, as indicated in the purple arrow, the depleted mice developed a rapid and fatal neurodegenerative disease characterized by progressive loss of motility and muscle strength. The disease was progressive, and all mice were affected, and death ensued. When we analyzed the CNS tissues by immunohistology, we could observe significant alterations in the CNS tissue. Compared to the control chimeric mice on the left panel, in which the monocyte-derived macrophages display microglia-like morphology, and in panel E, a normal amount of myelin staining, the depleted mice, in contrast, in mice in which the monocyte-derived macrophages lacked the TGF-beta receptor, they exhibited a progressive activation of macrophages over time. Seen here is the red staining in panel C, and this was associated with extensive loss of myelin, as you see here in panel E, by the loss of green color, and this was quantifiably significant, here showing the loss in myelin. This was also associated with the loss of nerve cell staining, as is apparent here at the loss of the red staining in panel F. Most importantly, in panel C, a number of very large macrophages in red was evident that I have ingested the myelin inside, which is green. So it seemed that once the monocytes infiltrated the microglia-depleted CNS, in the absence of signals via TGF-beta, they became pro-inflammatory activated macrophages and started tearing the nervous tissue apart, and it's this that led to the disease symptoms. When we investigated the transcriptomes of the different macrophage cell types using RNA-seq, we could observe many differences between bona fide microglia, that's to say the monocyte-derived macrophages, and the microglia-like cells in the TGF-beta receptor 2 knockout macrophages that were causing the damage. Many genes associated with inflammatory processes and phagocytosis were upregulated, which could account for the immunohistopathology we observed. In summary, we have developed a novel model of neurodegeneration that has aspects that are reminiscent of both MS and ALS. What is particularly interesting is that the damage to the CNS was focal, so that specific regions were affected. This indicates that there might be microenvironmental differences in the CNS that can affect cell functionality. So in essence, the microglia in one area of the CNS are different from those in another area. Such subtle differences in responsiveness to factors such as TGF-beta might thus be important clues to diseased, dysfunctional CNS states. Our new model provides us with a new system in which to test out our immunotherapies, and that will be our next research focus.